I don't know if we should start or we should wait for a few more minutes. 3 more minutes, and then we start. Okay, it's going to be 12:30 soon. We are going to start this session at 12:30. We are supposed to have 2 hours. But I know my friend and professor, Giuseppe Colla. He's a true professor. He will keep his troops in line and save some time for us. Am I right?
I will do my best.
As you can see, I have a lot of affinities for professors who are bringing commercial realities and connecting the dots. In that line, my next friend for this session as a session chair is Giuseppe Colla, who again, if you look at his signature, has 5 titles. So I will save some time and not go through 5 titles. That's why I didn't do the titles for John Crawford as well. He also has 5 titles. Am I right, John? So on that note, I will bring a very good scientist as well as someone who has brought a lot of science to reality and is also a very well-known professor, Giuseppe Colla, who is going to manage these 2 hours. All of you, please be ready to vote because all the speakers in this session are being judged for Bio Agro Disruptor Award. And as per the principle of this event, that this event is by the industry, for the industry, we will not like to choose the winner or loser. It is up to you. So please either go through the Hop-in web link or on your app and vote once they finish. As soon as they finish, please vote for them so we start the next speaker. The reason it is important you vote at the same time, so it's not that it becomes relative or you forget exact intensity or the understanding of their innovation. On that note, let us welcome Professor Giuseppe Colla to take the chair of this session. Thank you so much. Guys, come on.
Thank you very much, Roger, for this nice introduction. It's my pleasure to chair this section. And I want to start with a brief introduction of the section. If we can have the slides on the screen. Oh, okay, okay, I have the point. Okay, very good. So, in this section, we have 10 presentations dealing with the development of innovative products or approaches for developing products or studying the use of biostimulants. We have also presentation dealing with the understanding of the mode of action and the evaluating of agronomic performances. One focus of the section is the nutrient use efficiency and how we can improve the nutrient availability for plant uptake and nutrient use efficiency through the use of microbial and non-microbial plant biostimulants, especially for micronutrients and phosphorus that we know that are in many cases not so available for plant uptake. Another focus of the section is on improvement of abiotic stress tolerance to the use of plant biostimulants. Farmers usually attain half of the yield potential or even less in many field crops. This is due especially to the negative impact of abiotic stress factors. And biostimulants can help to mitigate the negative impact of abiotic stress tolerance, together obviously with the plant protection products. And biostimulants can also help to close the yield gap, increasing the food security. Talking about biostimulants, it's important to define the bioactive compounds and their mode of action. Biostimulant products usually are applied at very low dose in the field. Then the activity of biostimulant is mainly due to signaling compounds. These signaling compounds are able to modulate crop response at very low dose, altering specific gene expression patterns. But talking about signaling compounds, we have also a presentation dealing with the use of these molecules as a tools in integrated pest management to control pests by changing the behavior of these insects. The crop response to biostimulants depends obviously also by the interaction of biostimulants with the genotype and the environment. And there are many factors that need to be considered for optimizing the activity of biostimulant product: dose, formulation, frequency of application, timing, environmental condition, mode of action, and storage time and condition. And we will be able to see some of these aspects during this section. So now, before starting the presentation, I want to remind the expectation that we have from the speakers. They need to present clearly their innovations, focusing on especially on the unique features. It's very important to highlight the impact of innovation on sustainable agriculture and on the consumers and farmers, and to show the potential application of the innovation at commercial scale. Unfortunately, this must be done in a very short time. We have 10 minutes for each presentation. I recommend the speakers to use not more than 8 minutes for presentation in order to leave a couple of minutes for one or two questions. So now let's start with the first speaker. I asked to Vicente Navarro, from scientific researcher from University of Politècnica of València, to join the floor in order to present the use of semi-chemicals in pest management. Welcome.
Thank you.
Okay, slide. Okay, they are going to give.
Thank you.
Please be in time.
Well, good. Can you hear me? Yes? No? Very speak slowly? Okay, I will try. First of all, I would like to thank the organization for inviting me to give this presentation. I'm Vicente Navarro from the University Politècnica of Valencia. Wow. Okay, perfect. From here, from Valencia, and I want to talk about a case of a study of an invasive pest like we are knowing every day here in the world, in this global world. And this pest is a mealybug that comes from South Africa. It's the Rhodococcus aberiae. It's a special mealybug because it causes severe fruit deformation, and it's not usual in mealybugs, but it arrived here to Valencia region 15 years ago, and now it's a very huge problem for the growers of citrus. Of course, the first idea to control this pest is to use the classical biological control with these two species, Anahirus aberiae or Allotropa, the Rhodococci, but I'm working in pheromones. I work in developing pheromones, and what we thought is to fight against this pest using pheromones. But the problem is that the sex pheromone is not known. In South Africa, this mealybug is not an economic important pest, so they didn't study this pest, and we start with the study of this pheromone. When we want to apply control methods based on pheromones, we should know if the pheromone is known or not. If it's known, we only need to reduce the price to synthesize this substance. But when the pheromone is not known, we need a research to obtain this pheromone. And sometimes this pheromone is possible that doesn't exist, of course, because there are many species that don't communicate with chemical compounds. In our case, there is an evidence of the existence of these pheromones, and we need a research for the isolation identification of this product. This is a huge work of 2 or 3 years to isolate this pheromone, because we need to observe how the males and females communicate. We need to isolate the compounds that females are releasing. We need to kill all the males to obtain only virgin females, to extract all the volatiles. And at the end, with chromatography and nuclear magnetic resonance, we can identify the compound. And at the end, you have a compound like this, with this structure. And what we obtain is that using this compound, we have a very high attraction. But we want to see this attraction in the field. We need to place traps baited with the pheromone, and what we observe is this. One pheromone, one trap, sticky trap, just without pheromone, and the other with pheromone, and you obtain these catches. So at the end, we have the final compound with activity. So now we have the pheromone, but we should to choose which method we want to use with the pheromone to fight against this pest. Of course, if we have an attractant, we can do mass trapping or aggregation pheromone. But when we have a sex pheromone, the key point is the price of the pheromone. With a low-cost pheromone, we can do mating disruption with the sex pheromone, but when we have a very high cost, a very expensive pheromone like this, is not a straight chain pheromone or very easy to synthesize pheromone, so the cost is very high. We need to develop a method, in this case using a mass trapping strategy. The basic idea for a mass trapping strategy is to use a trap, in this case a sticky trap, like you can see in the sorry. Whoop, it's in the other direction, maybe not. Sorry, I don't know how to go back. Now. So, okay. Okay. The sticky trap, just placing one sticky trap per tree, we can reduce the population in the field and to reduce the fruit damage in the fruit. So with this idea that using this sticky trap and placing one sticky trap per tree, we can reduce fruit damage, and this is the idea. But we cannot replace the traps every 2 months, the pheromones every month to obtain this efficacy. We test the same device with other mealybugs, in this case Planococcus citri, another mealybug here from Spain, and we obtain a very similar result, a reduction in population and a reduction in fruit damage. So what we thought is to start a commercial development of this kind of traps. Not the sticky traps. We are going to use these devices with a company here in Spain. EPA is a company, Ecologia y Protección Agrícola, and we developed this kind of device with 2 tanks that contain the pheromone and the pyrethrin, natural pyrethrin. So we place these traps in the field, and we can see that many catches of males are obtained. Using this kind of device, we obtain many advantages. First of all, it's a very easy to apply trap. One trap per tree is very easy to hang. It's long lasting. We can release the pheromone during the whole year. We use natural pyrethrins to avoid other insecticides, of course. And the final advantage is that we can use the same devices for several pests. In this case, we are using the same device to place for Anideia auranti, for California red scale, for Planococcus citri, the mealybug of citrus, and for this new pest. So we are using the same devices to control all the samples. After we test, we talk with people of Bayer, and we have an agreement, a commercial agreement, to use this device in the field. And results after the first year of trials are, I think, are quite good. Because we have obtained a reduction of damage between 37 and 57% in fruits treated with this kind of device. I only want to thank Generalitat Valenciana for supporting this project, for economical support, and EPA, the company that has an agreement with us to develop this product. And thank you.
Thank you very much for this nice presentation. Is there any question? There is one question over there. Can you say your name and the question? No question. No question. Anyway, just one question from my side. I was thinking there was someone moving their hands. Where is used this product? Just in Spain, or do you have also applications in other countries?
Currently, it's only in Spain because this millibag has arrived to Spain, and it's only in the Valencia region. But we don't want, but we expect that this pest will be in all Mediterranean countries in not many years, because we can see how it has arrived 15 years ago and was very located in an area, but now it's expanding around all the Mediterranean area of Spain. And we don't know what is going to happen, but probably will be in all the Mediterranean area. We have a patent for all these countries because probably it will be spread. Yeah.
Okay, thank you very much again. Let's move to the next speakers. Now we have Daniela Kruse, Director of New Markets from GmbH, Germany. She's going to talk about multifunctional carrier fluids for enhanced storage life of microbial activity.
Yeah, thanks a lot. So my name is Daniela Kruse. I'm doing new business development for Evonik, and Evonik is a multinational company, a specialty chemical company. So we are not doing any agro-actives, but additives and also multifunctional carrier liquids. And I want to introduce our new multifunctional carrier liquids that enables enhanced shelf life and also higher field efficacy. So microbes and living microbes do have a lot of potential in agriculture. However, at the moment still lack often performance. This is due to several issues I would like to highlight here. So sensitive microbes, for example, fungal spores, have a limited shelf life and sometimes often use also cold storage. One reason for this is a non-optimal water activity. And another issue is the missing ease of handling. So they're difficult to formulate, and they are sensitive to moisture and pH. And to adjust this water activity, often living microbes are formulated in as solid products, and they very often have low dispersing properties in water. So the insufficient field delivery is an additional topic why the performance of microbials are lacking. So you lose parts of the products due to spray drift, to low adhesion and retention. You have a diminished contact on the leaf surface, and or the limited distribution in soil depending on the application. Taken together, you have a low or even an inconsistent field efficacy. We heard about this in the morning already. And due to this performance issue, the price to performance ratio is often considered as too high, and therefore we still lack the broad acceptance of farmers for these new technologies. And Evonik developed a solution for this. So this is not only for biostimulants, this is for a microbial-based biosolution. The breakthrough BP787 can enhance shelf life of microbial actives and in the different applications, so foliar, soil, and also seed applications. So the most important benefit mentioned here on this slide is the enhanced shelf life of these microbes. And we tested different ones. Here, these are the mentioned ones, Trichoderma, Bacillus, Clonostachys, and also Beauveria bassiana with increased shelf life. And we are currently running additional trials for additional microbes. You can use this carrier liquid, this liquid carrier liquid, to mix it with other Evonik breakthrough additives or silica products to get the desired properties in your final formulation. Breakthrough BP787 is a sustainable solution, so it's microplastic free, it's readily biodegradable, and no tox labeling. Mr. Braungart from Cradle to Cradle would call it intelligent chemistry. For seed applications, BP787 provides binder functionality with excellent dust reduction, so you do not need to add any additional binder. It enables hybrid solutions if you want to combine chemicals or micronutrients with your biological solutions. And I will show one example, and this is the reason why we are here in the biostimulant session. It improves the biological efficacy for biostimulants, biopesticides, and we also have enhanced seed germination in the seed treatment application. One important factor when I remember the talk in the morning, especially in North America and also Europe, are convenient solutions. And we have a liquid that has excellent rheology behavior, it's cold stable, and it enables low complex solutions. So it's mixable with water, you don't need to add any emulsifier, and yeah, we already patented this solution. So here I would like to show you how to do a microbial liquid formulation. You have, this is one example with Trichoderma hertzianum. This is 25% on silica. And use this biostimulant products and easily disperse these spores into the BP787 blend together with some additives. And we use this DA646 as a dispersant and the breakthrough S301 as a super spreader. The dispersing agent then this is the reason why you can easily steer the spores into the blend, and the dispersion concentrate is then easily dispersed also in the final tank mix, in the final water solution. You see this in the picture on the right top. The impact of the breakthrough S301 is an excellent soil wetting in this example, as it is a soil-applied biostimulant. So the formulation is storage stable for the biological spud. You see the colony forming units over 12 weeks at room temperature and with an enhanced storage stability test over 4 weeks, you have a constant colony forming unit count. And also physically, this formulation is stable. We have a water activity of 0.2, and this is perfect for Trichoderma, and we can adjust this water activity to the desired value. So we took this formulation and tested it in very unfed fertile soil that is limited in nutrients and especially in phosphates. And we treated maize seeds with this formulation, and in comparison to the commercial benchmark, we took the untreated control and the treated ones, we treated them twice. And in the end, we measured the fresh weight after 4 weeks. And as you see, we have a significant increase in fresh weight after 4 weeks. So in this slide, I would like to introduce some additives of our breakthrough portfolio, because as they are mixable with our multifunctional carrier liquid, they take care that we have a higher field performance. So let's look at this video. You have the spray droplet, and due to drift, you lose something. But also, if you do not have a proper deposition, then you have a bounce-off effect. So this water droplet that you saw here in this video, this would be lost under field conditions. Or you have a runoff. And if you add to this droplet 0.3% of our bio-based and biodegradable SP133, you have an excellent deposition and adhesion, and therefore the product stays where it has to be and where it can act. Okay, one additional video I would like to show. If you have never seen a super spreader active, then I think this is quite, so I'm a biologist and I think this is really impressive. So here you see the 50 microliter of water, and this is the same amount of water, only with 0.5% of our biodegradable super spreader. And you have a really great coverage on the leaf, on a hydrophobic surface, and so you have a good coverage and excellent adhesion. So coming to an end, with these high-performance additives, we can increase the field efficacy of biosolutions, of microbial-based agro-solutions, and the multifunction carrier liquid PP787 is for extended shelf life of living micro products, and it is a convenient liquid handling. And with a biocompatible breakthrough additive portfolio, we have an excellent dispersing of the spores and also an improved targeting as drift control, adhesion, retention, spray coverage, and uptake or penetration if needed. Thanks a lot.
Thank you very much. We have time just for a quick question. Is there anyone? Okay, please say your name and the question.
Thanks for the information.
I'm here.
My name is Mundar Yaqub. I want to ask you about, since this product is patented, maybe you can share with us the feedstock you use and the chemistry. Is it biodegradable? Is it organic? How do you classify it?
Yeah, so BP787 is a biodegradable solution. It's a synthetic product, so it's intelligent chemistry. It's not a bio-based product.
Thank you.
Okay, thank you very much. Let's move to the next speaker. I want to remind you that we need your vote, otherwise we cannot assign the award to the speakers. So please vote. And now it's time to listen the speech of Juan Antonio Hernanz, I'm sorry for my pronunciation, CEO of TBIO Crop Science, is going to talk about the intervention mechanism of TBIO functional peptide polycare on crop growth. Please.
Yep. Hello, everyone. I'm Juan Antonio Hernanz, Managing Director of TBIO Spain, company belonging to the group TBIO Crop Science, China. We are biostimulant manufacturers, and today we are going to talk about a product we developed called Polycare, which is a functional peptide. We are going to talk about the structure and characteristics. For the few that are not that much familiar, a peptide are a short chain of between 2 and 50 amino acids, in which well, they are linked by peptide bonds. Fewer than 15 is considered oligopeptide, and more than 50 is well, considered as protein as far as it's arranged in biological functional way. About the physical and chemical properties, well, should match this definition about molecular weight, 10,000 Daltons. Our product is between 3,000 to 6,000. Aphoteric property, actually this is quite important because sometimes the standard chelates, we will talk about that later on acidity environment does not work that good. Then the chelating ability, this is actually one of the key points nowadays is being talked when we talk about polypeptides. And solubility, very interesting, is higher near the isoelectric point, which as you also know, well, the solubility near to the isoelectric points is not good. Here, actually, this is going to lead us about what we were considering by the time we were developing our product. We choose some nitrogen species, amino acid, then some G families, in which the subfamilies are, well, different proteins, some of them with 700 amino acid, some other 400. But coming to the polypeptide, well, we have here the polypeptide as transporter, which actually is relevant for the nitrate uptaking. We have the oligopeptide, which actually is very relevant for the biological functions. And then what we have here, ABC, is a protein family which can be considered as a pump moving the substrates in and out of the cell. So actually what are we here talking about is like plants are not that different from human beings. So we need to eat, which is the polypeptide transporter. We need good digestion, which could be considered the oligopeptide. And we need also to have healthier cells. So this could be considered the ABC family of proteins. Now in the part 2, we are going to talk about the research with performance, in which relevant or key factors is which is the source, because there are different sources in which, okay, based on the standard definition or the standard results, animal hair is the one with a larger content, but we are going, or we focus, at least for our products, on the plant source. What we did is, okay, stimulating some effects. For this experiment, we used wheat, and what we choose, okay, different sources. One microbial fermentation, animal source, 3 different kinds, and plant source. So here are the results we obtain when we're talking about number of upregulated. Upregulated means that how do we enhance the result of the outer stimulus. So as we can see here, actually the podium is, we cannot see here. Okay. The podium is being done by the animal sources, while the microbial fermentation is following up. And the result of the plant source is that is not so good compared with another. But when we go inside of it and we make the analyze about how it's being divided, well, we got very nice results in terms of high temperature. When we are talking about stress, we are talking always about abiotic stress. So the result we obtained in high temperature with the plant source was really good. And actually, when we compare, like for instance, water stress with some animal sources, the result is really good as well. But coming to the topic of this presentation, which is crop growth, we can see here that now the one leading, making the gold medal, is the microbial fermentation, and that the plant growth shows very good result. Now here, part 3 is the commercial part of our company. We developed this product called Polycare. Well, poly means several, so why? Polycare actually is a peptide obtained by enzymatic digestion of natural proteins. Optimal molecular weight, as I mentioned before, was like between 3,000 to 6,000, and rich in active amino acids such as glycine, proline, and glutamic acid. Here again, somehow, we do like from the general picture to the one we concern. For instance, glycine have a very good property for chelation, while proline is a regulator of the internal function of the plants. And glutamic acid is just really good performance for plant growth. So again, it's like, okay, we eat, we have good digestion, and then, okay, we can get or expect better yield or better result. Now here we are going to briefly talk about, okay, how we develop this study with performance. Is like, okay, promote growth, regulate gene expressions. This part actually, with known technician's words, I like to explain with, for instance, switch and a bulb. Okay, you turn on, this is the external stimulus. You turn on the switch, and then maybe you have a wonderful bulb. You spend a lot of money, very powerful one, but the conductivity is not the proper one. So you cannot expect some optimal physiochemical results. You cannot expect very good light. You cannot expect the heat it should be given. So also something we were paying attention was about the root stimulation. In this picture, we can see the results we obtained in which the main root elongates. And this is like as easy as, well, we need good tools to have the upper part of the nice house. Here, okay, we can show also wide root system develop lateral roots and enhance root vitality. As we know, like a root can intake or uptake the nutrients 1 to 2 millimeters of distance. So, well, at that distance is where the part is being taken part. Similar thing happened, this one we did it with tomato, and actually you can see not only will affect the roots, also like the stem, the stalk, will be thicker and stronger. Promote flower growth. This is very, very important, and farmers, this is the part they like the most, because what does it mean, more flowers? Means, for instance, okay, this is cucumber or with tomato, whatever. One flower means one fruit. One flower more means, okay, one tomato more. So the farmer means he's going to collect more money. Frankly speaking, it's like that. And we can see the performance here, okay, for promoting fruit development with several parameters. We were taking into account fruit flavor in terms of brick, for instance, their color changing, fruited ripening in terms of trying to standardize. Well, the product finally result on the fields was like, okay, diameter and amount of flowers was improving. And, well, adjusted hormones. This is part of the properties of the oligopeptides in which we can show the result here compared with the lettuce. And, okay, how do we promote? Nutrient uptaking. Okay, promoting the nitrogen assimilation. We all know that, okay, nitrogen is a very relevant element in order to get better performance of the crop growth. We promoting photosynthetic carbon assimilation. We are helping the plant to breathe.
Can you finish because we are running out of time?
Okay. We're measuring here. Okay, last thing, only 1 minute more. It's like, okay, what we did took into account is like we can do not mix, for instance, fertilization with nutrition. We cannot mix, for instance, physiological problems with nutritional problems. So actually, we need kind of product which is a coadjuvant. It's not only focused on one function. It's not only focused about one stage of the crop. So we have to make our vision wider. So the conclusion we obtain with this product actually is like, okay, resistance to the adversity stress, promote the crop development, improve quality yield, and okay, synergy of other products. Actually, the way we also think in is a traditional Chinese medicine. We think like, okay, we prevent previous to the ill attack us in order to be healthier so we can cope or we can encounter better. It's instead of Pasteur was thinking like, I have a problem, I attack. So our idea is as, well, try to create those synergies and also by using our product, make the plant more resistant and prevent the abiotic stress. Thank you very much. Sorry for...
Thank you for your nice presentation and for these interesting results. I think we need to move to next speakers because the time is over. But anyway, during the lunch, if someone has questions, can contact
Thank you.
Okay.
Thank you.
Thanks again. Okay, let's move to next speaker. Romain Hanrion, Global Products Manager of Hello Nature. Is going to present a talk about peptide-based chelates, the power of synergistic action between biostimulant substances and micronutrients.
Thank you, Giuseppe. Hello, everybody. I will try to speak very well, but my accent, my French accent will be a difficulty, but I will try. So I will speak today about peptidic-based chelate, the power of synergistic action between biostimulant, substance, and micronutrients. Fertilization with traditional chelated micronutrients. Fertilization with microelements in chelated form has emerged over the years because of its efficiency. Despite the good chelating properties, synthetic chelates do not meet modern sustainability criteria of agriculture production, because of their many negative side effects, such as mobilization of contaminant metal ions in the soil, high persistence in the environment and plant tissue to their scarce biodegradability. Biochelation with vegetal peptide is a real true innovation. It has been discovered in our lab that some specific peptides have the ability to chelate micronutrients with the same effectiveness as synthetic chelates. The peptides involved in the chelation aren't only a carrier of metal. They also enhance the absorption of the element. They act as biostimulant. To boost plant metabolism, to limit stress caused by nutritional deficiencies, and to enhance quality traits. Here you have a small movie about the function of the peptide when they will biochelate a kind of metal ion. If I can try, yeah, it's okay. So you will see the amino acid will move to form a specific bond with the metal ion in order to biochelate any kind of bidental, metal ion bidental. So the advantages of the biochelation, it's 100% plant-derived, with a dual action means nutrients biochelation and biostimulating effect, with the effectiveness comparable to synthetic chelation, environmentally friendly, safe and sustainable, fully metabolized by plants who leaves no residue in the environment or in the crops. It's also able to act on different metal and not just as specific one. And we have on top of this biostimulant effect, which is a booster of plant metabolism improving of nutrients uptake, and the enhancement of quality traits. It's also able to maintain solubility of the metal at different soil pH, which can be a huge problem for the chelate. I choose three trials. We have made more than three, but I just choose three. One was made on grape wine in Champagne area in France. I think you know this area. So we try chelate EDDHA at 6% at a dose of 30 kilograms per hectare compared to our product, which dose is 11% concentration of iron at a dose of 16.36 kilograms per hectare. So we provided the same amount of iron in all the plots. Here you can see the iron concentration in the leaves. So on the left, the control, in the middle, the chelate EDDHA, and in the right part, our biochelate. So the concentration of iron was higher. In terms of yield, which is the most important for the grower, the yield difference compared to the control was 5% with the chelate and 16.70% for our biochelate. So finally, in this trial, we showed that we provided more iron for the plants, but on top of this, we found that we also increased the yield. That means that the biostimulation effect of our products. I also selected a trial made on tomatoes in greenhouses with hydroponic condition without recycling, made on rock wool substrate. The location was in Italy, and we tried two treatments, so chelated EDDHA at 0.3 grams per square meter per week, dose of application, compared to our iron biochelate. We use 0.164 grams per square meter in order to have the same amount of iron provided. The trial was a scientific trial with replicates of course and 6 plants per modality. On the left, you can see the foliar analysis and the iron content. That is the average of 3 foliar analyses. So 151.5 for the chelate EDDHA compared to 168.38 for the iron biochelate. So the concentration also is in trial, shown that a better concentration in iron in our modality. In terms of effective yield, based on 20,000 plants per hectare in greenhouse, was 110 tons obtained with chelate EDDHA compared to 115 tons with our iron biochelate. So it's an increment about almost 5 tons per hectare. That is also showing the double efficacy of our product, the dual action. And the last trial I wanted to show you, it's the zinc biochelate on corn. So the trial was made in USA, in North Dakota. So we have 3 treatments. So the control based only on starter fertilizer, applied at 46.8 liters per hectare. Second treatment was the same amount of starter fertilizer plus zinc chelate EDTA at 9% at a dose of 2.5 liters per hectare. And the last one was our zinc biochelate at 6% at dose of 3.65 liters per hectare. The plantation was made on 12 May and the harvesting was made in 2 November. So, sorry, too fast. Here you can see the foliar analysis in terms of zinc content. So you can see control on the left 4.5, 4.7 for the chelate EDTA, and for our product, the concentration in zinc was 6.7. And the yield, what is the most important for the grower, as I already said, it was almost 7 for the control, 7.02 for the chelate EDTA, and for the zinc biochelate, the yield was 7.26. So also an increment. So they are not the only one we have set up this year, or last year, sorry, but it was the one I wanted to show you. Conclusion, biochelation is a brand new technology, chelation based on selected vegetal peptides. The biochelation is 100% natural technology, environmentally friendly, harmless to living organisms, and respectful of the biodiversity. which leaves no residue in the soil or in the crops, matching the criteria of modern sustainable agriculture. Thanks to the presence of vegetal peptide, biochelation performs a dual action, nutrient biochelation and biostimulating effect that improve the absorption of the element itself, also boosting plant metabolism to obtain more yield. Trials performed in several crops show that biochelation effectiveness is comparable to synthetic chelation, and it also performs biostimulant activity which allow to reach higher level of yield. I would like to thank you for your attention.
Thank you, Romain, for this interesting presentation. Do you have any question?
Maybe it was clear.
Just one question. What type of nutrients can you complex with your technology?
So we can complex zinc, mangan, calcium, copper, and iron.
Okay.
All the bivalent cation.
Thank you very much.
Thank you to you.
Okay, let's move to next speaker. We have Puis Floris from Vitens, Netherlands. Is going to give a speech on natural alternative for synthetic iron chelates. So please.
Dear attendees from all over the world, thank you for taking the time to listen to my presentation for a complete new and circular vision on iron chelates. My name is Martijn Olde Weghuis, and I will give this presentation instead of Puis, so I'm not Puis, who told me last Monday he would not be able to travel to Valencia due to personal reasons. I am a business development manager for drinking water company Vitens in the Netherlands, and I'm working in the domain of circular economy. The valorization of byproducts from drinking water production are an important part of this domain. Vitens provides almost half of the Netherlands with clear and pure drinking water. It's a huge responsibility. We are 100% dependent on our groundwater sources, which are surrounded by farmland. Our task is to provide clean and affordable drinking water, now and in the future. Clean and healthy soil is crucial for both Vitens and the farmers in Vitens area. Both Vitens and PHC both Vitens and PHC share the goal to stimulate circular and sustainable agriculture and increase the use of residual products in agriculture. PHC stands for Plant Health Cure, is a 25-year-old company recently acquired by Den Oude Group. PHC is focusing on soil health and natural products for plant strengthening. Their vision is that only strong and truly healthy plants will produce resilient soils. Vitens makes impact by developing applications of byproducts in agriculture because our wells and our customers are surrounded by farmland. The aim is to replace or at least reduce the amount of conventional chemical or mining-related products used in traditional agriculture. This will have a positive effect on the quality, affordability, and sustainability of Vitens drinking water. What's not in it does not have to be removed. Vitens and PHC joined hands since 2012 to bring the valuable byproducts derived from drinking water back into the soil. from the ground into the soil. That's our circular motto. Production of drinking water is a resource recovery because besides drinking water, other natural resources are recovered during production. After extraction, methane dissolved in the groundwater is converted into electricity in a gas turbine. After that, iron oxides are filtered out as iron sludge, and at Spannenburg, which is a large production site in the peat-rich area of the Netherlands, this iron sludge product has specific attractive properties. It contains a significant fraction of fulvic acid that disperse and dissolve the iron oxides. We call this product ferro. More about this later. In the softening process, nice, round lime pellets are produced that find the market as a slow-release soil improver. At our Spannenburg site, the produced drinking water used to have a brown color, which is caused by the fulvic acids. We remove these fulvic acids nowadays with ion exchange technology. This produces a regenerate, which is rich in fulvic acids, that is further concentrated to a valuable product using an innovative process. The concentrate is marketed as biostimulant and soil improver by PHC under the name Fulvic. Vitens produces around 60,000 tons of natural byproducts per year, which is around 7 trucks per day. OLF, which stands for Organic Liquid Fertilizer, is a product from PHC based on Vitens ferro, which is the specific fulvic acid-rich iron oxide product from Spannenburg. It is a natural alternative for synthetic iron chelates to prevent iron deficiency in plants. We have started doing trials in 2015 on different crops in iron-limited systems, and the results are available on request. This picture explains the difference between regular iron sludge from the drinking water production on the left and iron sludge from Spannenburg on the right. Regular iron sludge consists of larger colloidal particles in suspension that will precipitate after a while. It does not contain any fulvic acids. The right graphic summarizes the research done on ferro by Wageningen University. Ferro contains fulvic acid that mobilizes and chelates the iron into fractions of different particle sizes, of which 20 to 40% is immediately available for plants. Last year, the Dutch company Keygene concluded a mode of action study based on molecular RNA analysis of the application of ferro on pak choi in an iron-free hydroponic system. Plants treated with ferro were far better developed and greener than the control, and sometimes even outperformed the reference iron chelate. The main conclusion from the RNA analysis was that ferro was capable of counteracting iron deficiency and that the mechanism is the same for synthetic iron chelates. It also seems that there is some kind of slow release effect taking place. Very interesting is that we also saw signs of biostimulant activity due to the presence of fulvic acid. Keygene found positive effects on root development, nutrient transport, and secondary metabolism. More info on this study is also available on request. So, the main conclusions and take-home message is ferro contains both natural chelated iron and natural fulvic acid. Both products are true innovations. Their use is intended to decrease the use of chemical fertilizers and minimizing the use of pesticides. Both Vitens and PHC provide products for the assignment to support agriculture with the sustainable goals. Thank you. Any questions?
Thank you for this nice presentation. Any question? We are getting close to lunchtime, so people is getting tired.
I have a quick question.
Okay.
Did you define the relationship between the PSC, Plant Health Cure, and the Viten? Did you do that in the beginning? Or it will be good to know for people what is the relationship between Plant Health Cure?
Okay, yeah. So PHC is a company that commercializes the byproducts from drinking water production. We have a long relationship with that company. They started with commercializing fulvic acids, and that became a success story because it's all the volume we produce is taken off by that company and sold to the market as a biostimulant and soil improver. Okay, thank you.
Thank you very much.
Thank you.
So let's move on to the next speaker, Nuria Sierras. Okay, she's coming from BioIberica. She's going to talk about biostimulant as herbicide safeners to improve crop efficiency.
Good afternoon, everybody. First of all, I would like to thank the organization for giving us the opportunity to present here today our recent results. Okay. Can you move like this? Yes. But first, let me allow us to introduce our company for those who still do not know us. BioIberica is a global life science company committed to improve the human, animal, and plant health. Our core business is the identification, extraction, and development of natural origin biomolecules, which are transformed into high-quality products for the pharmaceutical, veterinary, agrochemical industries as well. We are based on a circular bioeconomy principles, and we are integrally integrated. We are 100% bio-based raw materials, and our methods of obtention are specially designed to minimize the consumption of water, energy, and auxiliary materials. Before to present here our recent results, I would like to talk a little bit about the market framework of this kind of products. As you probably already know, it is predicted that by 2060, the global food demand will increase by 50%, which implies a rapid agriculture adaptation. What is more, currently the use of agrochemicals, understanding agrochemicals as fertilizers or pesticides, are highly inefficient. So with all this context, legislative decisions has been taken, and for instance, the European Commission has launched the Green Deal and Farm to Fork strategies aimed to reduce by 50% the overall use of risk pesticides by 2030. So with all this in mind, scientific researchers, we have the responsibility to bring solutions to develop more sustainable products. In this context, plant biostimulants has gained attention because they are a sustainable way to improve crops and quality. As you already know, they are applied into little amounts, into seeds, into the rhizosphere, into the plants, and they help to absorb, translocate, and use nutrients, and to overcome plant stress, abiotic plant stress. Which kind of a stress? For example, UV radiation, extreme temperatures, drought, salinity, and so on. But little attention has been paid on the stress provoked by pesticides, herbicides specifically. There are some scientific reports that state that this kind of compounds creates interferences into plants and affects their crop yields and quality as well. And from BioIberica, we have decided, or we have asked ourselves, how can we improve crop efficiency, trying to overcome this stress provoked by pesticides, specifically by herbicides. So in order to assess the features of these biostimulants regarding these concerns, we have carried out in controlled conditions a work in collaboration with the Universitat of Granada. We have tested one of our portfolio products, Terrasol foliar, with the herbicide imethamox at 4 milliliters per liter, alone and combined with our biostimulant. And we have obtained really good results. We have really nice data that we have already submitted to Plant Physiology Journal, and hopefully you will be able to read the whole work this Q2. But because due time limitation today, I will share with you just the key information. So here you can find experimental design. We have carried out 2 samplings. The first one was the same day of the treatment in order to assess fresh biomass, and then 4 days after the treatment to assess several parameters such as fresh weight, fluorescence, enzyme analysis, amino acid, phytohormones, phenolic compounds, in order to attempt to identify the mode of action of our biostimulant. Yeah, so results. One of the most obvious symptoms of the herbicide phytotoxicity is the loss of biomass. Actually, it is identified in several kinds of species, and it's used to determine the sensitivity or the resistance of a plant to this kind of stress. So they are then a reliable indicator in order to know if your biostimulant is a potential enhancer of this kind of, if it's a safener of this kind of stress. So here in the picture, you can see in treatment H, which is the herbicide treatment without the biostimulant, quite obvious symptoms like wilting symptoms, loss of vigor, the appearance of chlorotic spots on leaves, and less growth between nodes. Whereas these negative symptoms are specifically mitigated when the herbicide is applied in combination with the biostimulant. What is more, as you can see in figure 2, the fresh biomass is even 52% more when the herbicide is applied in combination with the herbicide. And the relative growth rate, which is another parameter related with the growth, it's nearly 6 times more when it's combined with the biostimulant. Okay, regarding the attempt to identify the mode of action of our product. As for example, glyphosate has been designed to inhibit the synthesis of aromatic amino acids. Imethamox family herbicides has been developed to inhibit the synthesis of branched chain amino acids, such as valine, leucine, and isoleucine. Therefore, ALS, the acetolactate synthase enzyme activity, is key in the synthesis of this kind of branched chain amino acids. Therefore, when the ALS enzyme activity is inhibited, that means it's a reliable indicator of herbicide phytotoxicity. So as you can see in figure 3, when the treatment is just the herbicide, this ALS enzyme activity is inhibited in comparison when the herbicide is applied in combination with our biostimulant. And what is more, if we analyze the amino acidic profile of the sunflower, you can see that these 3 amino acids are present in a higher concentration than when the herbicide is applied alone. And if we go deeper of this amino acidic profile, we can see that the precursors of these 3 branched amino acids, which are aspartate and alanine, are also promoted. And if we focus on, for example, glycine, glutamate, and serine, which are the amino acids that conforms the tripeptide glutathione, which in combination with glutathione S-transferase, which are a complex that helps to detoxificate the herbicide into the plant, are also promoted. With all this data, we have, well, quite a few data to affirm that the mode of action of our biostimulation is the promotion of this ALS enzyme activity, as well as the detoxification of the herbicide into the plant. Well, additionally to the study of the controlled conditions of our biostimulant, we have carried out several studies on open field last year in Romania with field crops, corn, sunflower, soya, and wheat. And here you can find the experimental design. And we have, again, assessed a lot of parameters, but due to time limitation, I will focus just on yield, that at the end of the day is what the objective of our work. So results, as you can see, in terms of kilograms per hectare, in all cases except for sunflower, we have obtained a significantly higher yield when the herbicide is applied in combination with our biostimulant than when it's applied alone. So as general conclusions for TerraSorb, TerraSorb, a biostimulant based on L-alpha amino acids and other ingredients, mitigates the herbicide phytotoxicity, avoiding plant growth reduction, increasing ALS enzyme activity, and key amino acids concentration related with plant optimal development and detoxification in sunflower. According to efficacy field trials results, TerraSorb diminished the negative effects of the herbicide on wheat, sunflower, and soya, and shows significantly higher yield increases. And therefore, the results open the perspective of using TerraSorb improving crops' capacity to cope with phytotoxicity and unwanted effects of herbicides. Thank you very much.
Thank you. Is there any question? One question over there. Please say your name.
My name is Brian Thompson. How do you differentiate between protecting the crop and protecting the weed from the herbicide? Is there any way to selectively protect just the target crop and not the weeds?
Repeat the question, please.
Your biostimulant will protect your crop from the herbicide.
Yeah.
It will also protect the weeds
Okay, yes.
from the herbicide. Is there a way to use it to only protect the crop?
Understood. Good question. Yeah, actually we have our own data about how the herbicide in combination with our biostimulant works against weeds. And we have the good data assuring that it works better. We have ED50 less. I mean, we need less active ingredient of the herbicide against weeds when we apply the herbicide in combination with our biostimulant. We have not identified the mode of action for the moment, but we think that it's more about physicochemical, physicochemical changes in the herbicide when we apply it in combination with our biostimulant.
Okay, thank you. Thank you very much. Let's move to the last speaker, Gorka, Erice, Global Technical Director of Atens. He's going to talk about the development of a biostimulant based on plant growth promoting bacteria associated with a mycorrhiza inoculum. Please.
Thank you, Professor Colla. It's my pleasure to be here today. The presentation I'm going to talk about is about PGP bacteria, plant growth promoting bacteria associated to plants. Let me talk about Atens before. Atens is a family-owned company. It was created in 1994, so we have a long history of 28 years. Atens started as a producer of mycorrhiza. It happens that nowadays we are the first producer of mycorrhiza in an in vivo system, together with a trichoderma producer in a solid-state reactor and bacteria in a liquid fermenter. We are very proud of our R&D department. It's the base of Atens. We have two inventions from the seed coating to bind the spores of mycorrhiza trichoderma to the seeds. and plant fiber with hygroscopic properties, which is essential to provide humidity to the microbes in the root zone and to the root itself. Well, R&D is the base of Atens, and this is the passion of the research. But we know that the method of production of a microorganism may condition its viability in the soil. So that's why it enters the passion for the quality. In the last 28 years, we have developed our own system to produce the mycorrhiza in a greenhouse, like you see in the picture, with our host plants, and we can recover the spores at the end of the season to prepare our formulation. And this is our solid-state fermenter for the trichoderma. But we also import the technology from other fields, like the pharmaceutical field, in order to mix the formulations and avoiding the contact of the microorganisms to any blade or any mineral oil, and also the machine to produce the tablet format products. There are three main ways to produce mycorrhiza. The first one is the open field, but you are open to get any contamination from pathogens that you can avoid if you use an in vitro system with a petri dish in the image. Well, but the spores that you can produce in an in vitro system, they are tiny spores that have less nuclei inside and have difficulties to colonize the roots of the plants. So that's why in Athens we opt to produce a mycorrhiza in a greenhouse with no contamination of pathogens, and we can control with the inner substrate the nutrition of our plants. That's why this is our greenhouse, and our host plants growing in tables separated from the ground. Here we can produce their high-quality spores, morphologically similar to the field spores of mycorrhiza, and the most important is the standardization, to get a product with viability, the product that you want to have the same good activity in the field for each batch. By the way, each batch is controlled by our quality control, but also with the IRTA, the Catalan Institute for Agronomic Research, as a second quality control. The advantage of this system is that together with the mycorrhiza, we let some bacteria to grow with. In fact, we have identified up to 15 bacteria which has plant growth promoting activity. Some of them are capable to fix the nitrogen in the atmosphere. Some others can solubilize the phosphorus or can produce auxins or siderophores for iron uptake from the soil. Well, this is a big advantage because we have combined two of these bacteria, two Bacillus megatherium strains, 06 and 77, to produce a new product which is called Bactrium. This product has a final concentration of 10 to the power to 10 colony formation units per milliliter. The idea behind is to combine these two bacteria that can solubilize the phosphorus that the farmer has applied in the previous campaign, and also they can produce siderophores for iron mobilization. This is the display of our liquid fermenter for bacteria production. Here, the bacteria grows to a fully population, and then we can induce the sporulation of such bacteria. So the final product is going to be liquid with no lyophilization, no filtering that can compromise the viability of the bacillus. One of the smart ideas to combine the phosphorus and the iron is because their availability in the soil depends on the pH, and the availability of phosphorus does not overlap the availability of the iron. Both phosphorus and iron share a common system to be uptaken by the plants. That's the acidification of the soil. That's why they can interfere, and the availability of phosphorus can condition the availability of iron and vice versa. Sometimes, when you have a low phosphate concentration, maybe the iron can accumulate negatively with toxicity in the roots, and that can condition the plant growth. On the other side, when you have a low concentration of phosphorus in the soil, well, that may aggravate the iron chlorosis in the leaves. Finally, I can show you some of the results we have obtained using Bactrium. Here, we use 2 different seeds of corn and soybean, and a treatment of Bactrium of 200 milliliters per 100 kilograms of seeds. Well, with these systems, we have promoted the early germination of soybean up to 6% and corn up to 14%. We didn't affect the later germination, but what happened during this short period of time promoting the early germination? Well, we have conducted a metabolomic analysis to certify that these plants have increased degradation and utilization of the research protein, as well as they have higher content in anthocyanins, and also the purine nucleosides, which is a statement of this greater cell division and also the nitrogen assimilation that was promoted by Bactrium. In another trial carried out in blueberry, in a cultivar which is named Colla and has lots of problems of cracking, well, the application of Bactrium led to a higher weight of the fruits, 35% higher, and the higher size of such fruit, well, 6%. So we have not only a bigger fruit, but a more dense fruit. A very nice result in the increasing of 37% of the ferments, avoiding the cracking in the blueberry fruits, together with a higher content of sugars and no affection in the acidity of the fruits. And just to finish, a trial that we conducted using lettuce in Italy in 2 different locations, in Tucsia and Tarquinia. Well, in these trials, what we have seen is that the application of Bactrium induced the higher concentration of chlorophyll in the leaves. In this case, even a 7% higher concentration of chlorophyll, which led to a higher fluorescence, that a higher capacity of the plants to avoid the photooxidative stress. And regarding the production, we have seen how the application of Bactrium increased in 9% in the case of the test number 1, and 4% in the case of test number 2. So just to summarize the effects of Bactrium in lettuce, we have increased growth rates, we have the increased chlorophyll content, even upgrading the efficiency of the photosystem too. That's the dissipation of that excess of light energy, and with the greater average weight of the lettuce, which is a very great performance test for the farmers. And the last slide is just to thank you very much for your attention today.
Thank you very much, Gorka, for this nice presentation. We have time for one question.
People is hungry.
No question. I have a question for you. How do you...
John has a question.
Oh, sorry. I didn't see your hand.
I was hiding.
Okay, go ahead.
I noticed there weren't error bars on the graph that you showed for the improvements for the lettuce. Are these increases significantly? Are they statistically significant?
Significantly, do you mean after a statistical test? Yes, some of these increasing are statistically significant. Some of the parameters, they have a high variability, but the increase in the growth is very consistent. It's one of the consistent ones.
Okay, thank you again, Gorka.
Thank you, Professor.
Okay, this was the last speaker. So before closing the section, let me say a few words as conclusion. We have seen how much science and technology is behind the innovations clearly presented by the speakers, and I want to thank all of them, especially for being in time in the presentation. We have seen that to be successful is fundamental the integration of different approaches, including omic technologies like transcriptomics, metabolomics, in order to identify the signaling compounds and to clearly understand the mode of action of the products. However, we cannot forget the importance to go deep in studying the interaction between biostimulants, genotype, and environmental conditions to well-designed agronomic trials. Moreover, it's also necessary to study the integration of plant biostimulants with other inputs like plant protection products and to take advantage of the synergistic interaction. The final goal is to develop functional plant biostimulants able to solve specific problems in specific growing environments. This can contribute to reach the goal of producing more with less, the so-called sustainable intensification. I hope that the section meet your expectation, and I wish all of you a good lunch and networking activity. I don't know if Roger has some announcement to do.
I just want to add some housekeeping things. We will be back at 3:15 for a very interesting session chaired by our own Pam Marone. And that session is going to have one keynote speaker, and the rest of the speakers will be all paper presenters. And I totally echo the science, what I saw and heard, and the time management, except one, was fantastic. We were watching the stopwatch, and it was always on dot. So thank you very much for keeping that time. And John, the project that is working now, but I'm sorry it didn't work for you, the pointer. So we'll see you at 3:15.